Traveling inward through the lipid boundary of a mammalian cell, we enter the dense, spherical environment of the nucleus. This compartment is microscopic, spanning a diameter of approximately 10 to the -6 meters. Contained within this single micrometer of space is the full human genome, a massive dataset consisting of 6.6 billion base pairs. These pairs are not packed at random. Each pair requires a fixed physical distance of exactly 0.34 nanometers of clearance from the next. When we multiply the 6.6 billion base pairs by that 0.34 nanometer spacing, the resulting linear polymer reaches a total length of 2.2 meters. At this scale, the genetic material in a single cell is a human-sized object. The spatial paradox is absolute. The cell must house 2 meters of physical cable inside a container 1 million times smaller than the cable itself. Managing this data load requires a rigid hierarchy of electrostatic packaging that begins at the atomic level. To understand how this packaging works, we have to look at the polymer from the bottom up, starting at the sub-nanometer scale. The fundamental monomer unit of this system is the deoxyribonucleotide. The central chemical hub of this unit is a 5-carbon pentose sugar called deoxyribose. At the 1' carbon, a nitrogenous base attaches via an N-glycosidic linkage. At the 5' carbon, a phosphate group is linked through a phosphoester bond. These nitrogenous bases come in 2 sizes: the larger double-ring purines, adenine and guanine, and the smaller single-ring pyrimidines, cytosine and thymine. A single nucleotide is just a precursor. The genetic chain only begins when a second nucleotide joins the first. The units join through a 3' to 5' phosphodiester linkage, where the phosphate group of one sugar locks on to the hydroxyl group of the next. Because each unit joins in the same orientation, the chain possesses a physical polarity. One end has a free 5' phosphate, while the other has a free 3' hydroxyl. This creates a rigid directional sugar-phosphate backbone, from which the data-bearing bases project outward into space. With the nitrogenous bases projecting away from the backbone, the genetic sequence remains physically exposed to the cellular environment. Watson and Crick utilized physical models like this one to demonstrate how a second complementary strand could shield these bases. For the 2 strands to fit together geometrically, the second chain must be inverted, running in an anti-parallel 3' to 5' direction. The pairing between these strands is governed by strict rules. Adenine only maps to thymine, and guanine only maps to cytosine. These pairs are held together by hydrogen bonds. We see 2 bonds forming between adenine and thymine, and 3 bonds locking guanine to cytosine. Pairing a bulky purine with a smaller pyrimidine ensures the horizontal distance between the backbones remains constant. As these bonded base pair layers stack on top of each other, their internal geometry naturally twists the structure into a right-handed spiral. One full turn of this spiral covers a pitch of exactly 3.4 nanometers. 10 base pairs fit within that single turn, confirming the 0.34 nanometer spacing per base pair we established earlier. This double helix geometry is the inevitable result of hydrogen bonding and base stacking forces reaching a mathematical equilibrium. While this helical structure is stable, we are still left with a rigid 2.2-meter cable that must be compressed. Simple loops are sufficient for prokaryotic DNA, but the scale of the human genome requires an active mechanism for massive volume reduction. Compression is difficult because the phosphate backbone is highly negatively charged. As the strand tries to fold, these charges repel each other, preventing tight packing. The cell overcomes this by utilizing histones, a class of positively charged basic proteins. This positive charge comes from a high concentration of 2 amino acids, lysine and arginine. The negatively charged DNA is attracted to the positively charged histone core, wrapping tightly around a unit of 8 histone molecules. This wrapped structure, the DNA and its histone core, is called a nucleosome. Each individual nucleosome secures approximately 200 base pairs of the DNA helix. This electrostatic attraction neutralizes the backbone's internal repulsion, allowing the 2.2-meter polymer to finally begin folding. We can observe the results of this molecular folding directly using high-resolution electron microscopy. Under the microscope, long chains of nucleosomes form a structure called chromatin, appearing as a repeating beads-on-a-string pattern. This string is coiled and condensed further by non-histone chromosomal proteins, forming the metaphase chromosome. Even here, packaging density is not uniform. Loosely packed chromatin regions are known as euchromatin. Densely packed regions are called heterochromatin. The physical density of this packaging directly determines the biological function of the DNA. Because euchromatin is loosely packed, it is transcriptionally active. The cellular machinery has enough physical space to read the genetic sequence. Conversely, the heterochromatin is so tightly condensed that the genetic information is physically inaccessible and remains inactive. The molecular basis of inheritance is a mechanical system where sub-nanometer chemistry controls macroscale packaging, regulating 2.2 meters of data through structural density.